Hello and welcome to the Marine Community Ecology course. If you are taking this course as part of the UT Marine Science Institute MNS 352 or 382, then you can stick around for the first half of this lecture recording. If you are taking this course independently, however, you are welcome to skip through to the later sections of this presentation, just because it covers some of the structural elements that are unique to having this course being taught within UT Austin's systems. So what we're covering today is an overview of the general course structure. We'll go through some housekeeping things. We'll have a short introduction to R, a little bit of background on me and my research, and then a primer to community ecology. The course structure is as follows. So we are meeting up on Mondays from 10:30 to noon for our lecture. So this is for the same slides that you are looking at right now. And then we're meeting up on Thursdays from 1 through 4, and this is for our lab. Our class time is generally in the UTMSI admin wing video classroom or on Zoom, on the provided Zoom link. I'm your instructor, and my email address is right there if you ever want to reach out to me. My office is in the Marine Science Institute in the main research building 305, and if you want to do office hours, they are by appointment, in person, or on Zoom. This course has two sections. It has an undergraduate section of students here in Port Aransas, and then a graduate section with graduate students either in Port Aransas or potentially in Austin. There are distinct learning outcomes for undergrads and graduate students. So the first slide here has the learning outcomes for all students. It is to gain a thorough understanding of ecological theory as it pertains to community ecology, to acquire or improve, depending on your status, your computational skills to explore and analyze and visualize data in R, to express yourself in scientific writing. This one's very important to me. Generally speaking, I think writing is one of the most useful skills you can hone as an aspiring scientist. To present your work and follow or contribute discussions. So this is verbal articulation of scientific content. Then to survey, sample, process, and analyze marine and estuarine communities in the field, as well as to learn how to assess your own progress and growth in a non-traditional grading format. And more on that in just a little bit. The learning outcomes for graduate students are three extra ones. First of all, I want you to gain advanced skills in R, where you can troubleshoot and help other people troubleshoot their own issues. I want you to develop some leadership skills in research, meaning a capacity to steer discussions, make decisions, find solutions in delicate tasks, and also to synthesize research outcomes in a brief and very compelling pitch to a journal using a cover letter, an abstract, and a couple of figures. The different elements of this class are lectures and labs. The lectures are 80 minutes. The slides will be on Canvas and on this website 30 minutes prior to the lecture, just depending on how organized I am. And the labs include coding labs, where we do class exercises. They include fieldwork and they include laboratory processing. So if you are not taking this course as part of UT Austin, then you are unfortunately missing out on the fieldwork and the lab processing, but there are some suggested activities on this website that you can follow. The TA and co-instructor for this course are Christina Marconi and Dr. Chris Hemmingson. As you will notice, they are not here right now. That's because they are in Australia currently doing fieldwork. They will join us in a couple of weeks' time. Okay, a little bit about the resources and policies in this class. So Everything is, of course, on the UT Canvas website as well as on this companion page. So generally speaking, I would recommend you check Canvas regularly, ideally sometime before you go to class, just to make sure you've gotten all the announcements and everything. There are some other resources on the syllabus as well that you can consult. And then this companion page, obviously, if you're watching this recording right now, then you will have found the page. Feel free to browse the page. If there are any issues on this page, it's constantly a work in progress. Do let me know and I will attend to it immediately. A few words about policies. I'm not a stickler for any of the policies necessarily, and there's a whole lot of text and a whole lot of excitement around them. Generally speaking, the important part is that I will not accept plagiarism. I am a responsible employee and I will report all Title IX incidents. And perhaps one thing that is worth pointing out that concerns the use of artificial intelligence, well, suit yourself. You are the one paying for this class, not me. If you choose to use AI to complete the assignments for this class, then be my guest. Just realize that I will know, because you always do. And it's also just not helping you learn very much. But I'm happy to discuss this in more detail for some of you that are desperate to use AI in some capacity. But again, I will not be the one forbidding you to use it. And then finally, I do want this to be a safe and inclusive learning environment. So please respect each other, each other's contributions, provide constructive criticisms without judgment. I will honor any request to change your name or pronouns from those that are listed on the official course roster or on Canvas. So please let me know if that is the case. And then just a couple of words, since we are sort of digging into the depths of community ecology and its history, you will notice that the history of community ecology is dominated by a certain demographic group. That was the group that was privileged enough to do a lot of ecology all throughout sort of the early and mid 20th century. I am doing my best to provide different perspectives where possible. Just realize that there are a lot of historical inequities that have led to a concentration of work in certain demographic groups in the early stages of ecology. Okay, a brief overview of the actual course content. So there are 4 modules in this course. They essentially are an introduction to community ecology, a new-ish theory of ecological communities that tries to pull a lot of that early work into an overarching framework. We're going to then look at the empirical evidence for the processes that are part of that new framework in marine systems. And then finally, we'll finish up with some of the most current research challenges for marine communities in the 21st century. A quick word about the labs. So a lot of our labs are going to be coding labs, which means that you may have come down here and expected fun fieldwork during all of the labs. That is not going to be the case, although I will wager that coding can actually be just as much fun as going into the field. I may be on an island with that opinion, but hopefully it'll be enjoyable and useful for you as well. Speaking of coding, let's have a brief word about R, which is what we're going to be using for our coding exercise here. So what I want you to learn really is to execute basic commands in R, to load and use packages, to manipulate and analyze data, plot the data and their uncertainty. You will run some basic simulations as well, which I think is very useful, and then you will run some multivariate ordinations, and maybe if we have time, also some more. For R, the first module is just going to be an introduction, how to process data using tidy language and the tidy workflow, and how to visualize them. Pay attention and try to sort of stay on the ball during that first module, because it really is the basis for everything else you'll be doing in R in this class. The second module is going to be simulations of ecological processes. It's a fun one. I think it's really empowering to play with simulating numbers in R and to plot them to see how different processes would shape an interaction. The third module will be multivariate analyses and potentially some trait-based analyses, which kind of fall into the same framework. And then the fourth module, which is really sort of spaced out throughout the second half of the semester, is to analyze and visualize your own data. Why are we using R? Well, first of all, it's free. Nobody's making any money of you using R, which is phenomenal. It's the most commonly used platform in ecology and evolution, at least as far as my experience goes. Some people will use MATLAB or Python to do their work, but by and large, I think R is the most common one. It's open source, it's community built, which means that you will mostly be working with these building blocks called packages that are made by researchers and people who contribute to this community. We have built a package which allows you to color your plots based on the colors of coral reef fishes, which frankly is not the most useful package in the history of packages, but it's fun anyways. And the other thing about R is that it forces a basic understanding of statistics, unless some of the like drop-down menu statistics platforms that have an extensive GUI. And it helps you gain programming skills, which I think in today's market are extremely valuable. Upfront, R can be challenging. R has quirks, R has challenges. It can really be hard work to do stuff in R. And to this day, I've been working with R for more than 10 years, and this is like the perfect encapsulation of how I feel like I've progressed in R. I still bang my head against the wall occasionally when I can't figure out why something doesn't work. But then sometimes you also feel extremely empowered when you realize that, hey, actually, I do know my way around this and I can code. For you, the first steps will be to download R. I strongly also recommend downloading R Studio, which is basically an interface that runs R. And then if you are completely new to R, the Swirl package, I think, offers a really nice introduction. But you can essentially go through these steps by following this link right here. This is to the companion page, the platform that has all of the coding stuff. If you just navigate to the overview section there, you will get a nice primer for how to do your first steps with regards to R. Okay, the other labs are going to be fieldwork and lab work. This is a short video that I'm not going to play in this lecture. You can navigate to YouTube and find it yourself. But essentially, our first bit of fieldwork is going to be about shorebirds and other coastal birds. We are going to be surveying these communities here in Port Aransas, and then we will work with the data we have obtained. And the second bit of fieldwork that we're going to do is with fish. We're going to catch fish, we're going to work with these fish, and it's going to be a whole lot of fun. Okay, great. Now this is the fun part. I will not be providing any grades for you in this class. That might be a fairly unusual thing to do, and it may induce some anxiety for some of you because you feel like it's a trick. It's not. As a matter of fact, I think that it is something that all of academia should move towards. The basic theory behind all of this is that if I am the one grading you, then I am making myself the sun in your universe, which means in other terms that I am providing the material, I am setting the expectations, and I want you to fulfill my expectations for learning and growth. And given that you are the student, I don't actually necessarily think that that is the right thing to do. In turn, what I'm hoping to achieve here is for you to be your own son, to essentially make this course part of your universe and to guide your own learning and growth. And by not having instructor provided grades, I'm hoping to free up this element in you, or this side of you that actually wants to learn and wants to grow, rather than feeling like you have to in order to get a certain grade. Now, I know this is a little ephemeral and a little esoteric. In practice, how does this work? Essentially, we are going to build a system of trust between you and me, and that system of trust is formalized in what's called a learning contract. Now, that contract has a promise from my end, which is that if you simply submit all of the deliverables, all of the assignments, homework, and so on and so forth, you're going to get a B- at the absolute worst. So that is my side of the contract. However, I imagine that most of you will want something more than a B-, and that essentially then brings in your side of the contract, which is that you will set your own goals and expectations for each of the learning objectives for the course, considering where you're coming from and where you want to go. And then at 2 times throughout the semester, one time at the midway point and one time at the end of the semester, you will reflect on your own learning and growth and achievement of the goals you have set. And at the very end, you will use that self-evaluation to essentially propose a grade for yourself, because UT requires me to still enter a grade. You will propose a grade for yourself, and 95% of the time I will enter that exact grade into the grade book, and that's done. I will reserve the right to change that grade, either for the worse or for the better. So there is that element. Rest assured that in the vast majority of cases that does not happen. But if I fundamentally disagree with your assessment, I will reserve the right to change it. Again, these are the learning outcomes, and essentially what I'm going to ask you to do is to set up this contract in a format that is similar to this. You are welcome to amend this. This is just a template, sort of just to give you some structure on how you might draft this contract. So on the far left, you have a learning objective. So these would go 1 through 6, the learning objective of the course. And then you would have a column that is your goal and your foundation. So for this one, for example, the learning objective is gain a thorough understanding of ecological theory. And say you are interested in pursuing a graduate degree in ecology, and you really want to exceed expectations in this one. And you would add something like, I have a good foundation in ecology, but because you want to do something with ecology sometime down the stretch, you really want to knock this one out of the park. And then you can sort of devise some actions on how you might achieve that. So some of them are obvious, like complete all the readings that I'm assigning. Well, if you want to go above and beyond, then you certainly should do that. You want to stay engaged, and then you can also think about some really quantitative ways of how you might be able to go above and beyond. So for example here, and keep in mind these are just examples, it would be read at least 2 extra papers a week, spend 1 hour per week reviewing class material, revise written assignments to incorporate feedback, and challenge yourself to ask at least 1 question during the graduate student symposium that we're going to have. Again, there are lots and lots of opportunities here to come up with these quantifiable, tangible actions. The important part is the more quantitative they are, the easier it is going to be for you to assess yourself. The assessment then is for the mid-semester and the final, and you can read these in your own time. again, these are just examples. And then on the final assessment, you're going to consider all of this and you're going to come up with sort of a percentage score of how you think you did on meeting your goal based on what your expectations were. And then you do that for all the 6, or for the graduate students, 9 learning objectives. And at the end of the semester, I'm going to ask you to consider your assessments, and ask for your grade with a short justification. These are some of the actions. I've just listed them here. Again, be creative. There's lots and lots you can ask or engage in to do this. They're just a list of potential actions you could take. One thing I want to point you all towards is the concept of metacognition, which is a theory of assessing your own learning in a better way. So being aware of your own learning and growth. I think it's a really important skill to develop, especially if you are planning to be in academia in the long run. Okay, the final thing for all of this is that I want you to talk to me about your learning contract. So I want to know what your expectations are, where you're coming from, what you want to do, and how that guides your own learning contract. That will be best in person, so I'm asking all of you to at some point come talk to me in the beginning of the semester over the first 2 weeks. I'm generally in the office and happy to take ad hoc meetings unless I'm in another meeting, so just come on by. There's no need to schedule unless you really want to. And I am really quite nice, so don't be scared. And if you think that's a lot to ask, then just think about how I'm feeling when I have to meet with every single one of you. Okay, assignments. Just because there's no grades doesn't mean that you don't have to do work in this class. As a matter of fact, I would wager that if you want to exceed all expectations in this class, and if you want to get the very most out of it, this is going to be one of the hardest and most challenging classes you will take. For both undergraduates and graduates, there will be 10 homework assignments throughout the semester. There'll be 2 self-evaluations that I just mentioned, the mid-semester and the final one. And then there's 3 separate projects, 2 written assignments and 1 oral assignment. For the graduate students, because they are much more advanced now, they have 2 extra assignments. One of them is a presentation for the first project, and then the second one is a paper pitch, which is essentially something you would write to a high-impact journal if you are trying to submit your paper there. The homework assignments are about an hour to 2 each. The details on each homework assignment are provided in class, and they need to be fully completed and submitted by 9 a.m. on the due date. The first assignment is what I call a paper critique. And essentially what this is, it is a critical evaluation of a seminal theory or hypothesis in community ecology. The foundation of this assignment can be done as a group activity. So I'm asking you to essentially pick a theory from a variety of papers that I'm providing, and to take that paper, read the theory, and then sort of do a follow-up search on how this theory has fared in the literature. So as an example, I could use the theory of island biogeography and then assess how has the theory of island biogeography fared in follow-up work, follow-up research. Has it been useful? Has it been proven? Has it been disproven? And importantly, that does require you to find some literature, right, that either supports it or refutes it. And that's the part where you can act as a group and you can discuss the different papers as a group. But keep in mind that each paper, so each critique, is written individually. I will provide more information on this assignment later on, but essentially we're talking about 3 pages during which you introduce the theory, present some evidence for and against that theory, and then finally come up with a conclusion that either supports or refutes that theory. So essentially you get to be the judge of, is this theory garbage or is it good? The second project is a research paper that you will do on the survey data from the coastal bird communities that we will collect in the field ourselves. It will also integrate some historical data from this area, and it's essentially a classic research paper with abstract, introduction, methods, results, and discussion, about 5 pages. Again, more information on that to come. And then the third project is going to be group-based data collection on coastal fish communities. And then we'll do some laboratory processing of these fishes, and you will do some data analysis and visualization. And then you have group presentations that you will do sometime towards the very end of the semester. The graduate students, as I mentioned, they have a couple of extra things. You have a presentation about your paper critique. So you essentially get to stand up in front of an audience and defend or trash your theory that you've chosen to write your paper about. And then you have this paper pitch, which is essentially a cover letter, a title and abstract, and a couple of figures for submission to a journal. And this is based on the data we're collecting during the second set of field surveys. All right, so a little bit about myself and my research before we dive into the actual topic of this class. I grew up in Munich, in the foothill of the Alps in Europe, and did my bachelor in biology at the University of Innsbruck in Austria, in the Alps, so not a lot of marine science there. Then went on to do my PhD at James Cook University in Australia, working on coral reef fishes, especially herbivorous coral reef fishes on the Great Barrier Reef. I then did 3 postdocs with the Smithsonian at Simon Fraser University in Canada and in France. And since 2021, I am an assistant professor here at the Marine Science Institute with UT Austin, and I'm having a mighty fine time. My lab generally works on these 3 really broad topics: marine biodiversity, ecosystem functioning, and global change. If you want to learn more about our research, you can check out our website. But in principle, here's what most people think I do most of the time, which is do fieldwork in some really nice tropical, warm, clear water, collecting fish, looking at fish, counting stuff underwater. And I do that sometimes still. In reality, the top right is probably the photo that encapsulates best what I do these days, which is to stare at my computer, answer emails, and sign things that I don't really want to sign. But such is the career path of the tenure track faculty member. It's been a fun ride so far, and I've enjoyed every minute of it. And if you ever want to talk about career stuff in marine science, feel free to come talk to me. Most of the time I deal with these people here. This is my lab, or a good chunk of people that are in my lab. They're a joy to work with, and if you see them around, feel free to introduce yourself, say hi, talk to them about their research. But yeah. All right, now let's dig into the actual topic of the class. And the creature I want to open this with is this guy right here. This is a Steller's sea eagle, which is, for all I know, the heaviest and largest eagle in the world. It's a beautiful bird, and the Steller's sea eagle at some point showed up in Maine in January 2022. and thousands of people flocked to Maine to see this bird. And the reason why that was so attractive is because, generally speaking, Steller sea eagles are not supposed to be in Maine. Their native range is in northeastern Asia, so they occur in Japan, Russia, Kamchatka. And this one individual clearly had gotten very, very, very lost. Allegedly, it was also sighted at some point in Victoria, Texas, just about an hour from here. That hasn't been confirmed, but we certainly know that this eagle was straying far from where it was supposed to be. But the question really here is, who says that the bird wasn't supposed to be there? And what determines where a species actually occurs? And that is a question that is sort of at the core of marine community ecology, because the species are what makes the community, right? So we're always interested in understanding why certain species occur here but not there. A great example for this are the orders Sirenia, which are your dugongs and manatees or sea cows, which some 50-some million years ago apparently derived from land-eating cow-like animals, took to the oceans and made a new home there. And especially in the Atlantic regions, we have three different species of manatee: the West Indian, the African, and the Amazonian manatee. And then in the Indo-Pacific, we have the dugong. And for all intents and purposes, these things look exactly alike. They're all the same thing from a biological perspective, yet they are different species. And they are different species because they are tied to these coastal waters. And when our continents sort of drifted apart and sea levels fluctuated, these populations ostensibly got separated at some point and eventually settled into their new geographic range and became reproductively incompatible with the others, and hence we have different species. So now we have 4 species that occur in different parts of the world. And a great counterpart to this are humpback whales. So humpback whales occur pretty much anywhere in the world, from the tropics to the poles, in the Indo-Pacific, in the Atlantic. They're just about anywhere. And the reason they haven't diversified into different species is because they are tied to the pelagic environment rather than the coastal environment. So for manatees and dugongs, obviously they require shallow, productive coastal habitats where lots of light gets to the bottom, seagrass can grow, and they can forage on the seagrass. But for humpback whales, they rely on pelagic resources, krill and small fish, that don't tie them to the coasts. And as a consequence, their populations evidently have, stayed connected throughout millions of years, and they have not diverged into different species. So there you go. There's one example of how biological traits in different species interact with the shape of our world to explain why some species occur where and others occur, for example, everywhere. Another example of species that occur in some places at some point in time but don't in others is this guy right here. This is Dunkleosteus, a placoderm that ranged very, very widely throughout the world's oceans some 300 million years ago. However, you are now very unlikely to see a placoderm. As a matter of fact, you will never see a placoderm in the wild because they've been extinct for quite some time, and hence you will not encounter them anymore. Other things that shape why species occur someplace but not another is, of course, climate. So, for example, tropical coral reefs harbor their own species that are adapted to coral reefs, whereas the poles, the Arctic and Antarctic, have a very different suite of species that can thrive there. Within sort of latitudes and regions, a major determinant of what you're going to see in terms of the species, is the underlying habitat. So a coral reef will harbor a very distinct or very different suite of species compared to a soft bottom habitat, just by providing a lot of structure and food. And then anthropogenic impact is also going to impact this. So for example, a reef on the left that has undergone some degradation is likely going to look very different from a reef that is thriving and harbor different species that cannot exist on a degraded reef. And one thing that we really want to keep in mind when we think about community ecology is that life is really the one thing that makes Earth unique, at least in our solar system and as far as we know, in our known universe. And life really operates from the spatial scale of biological cells to the entire planet. And if you sort of scale up through all of this, you'll find that community ecology basically sits in the middle of all of this, which really makes it quite interesting from a spatial scale point of view. What I mean by that is that communities can exist at these vastly different spatial scales. So, for example, a drop of water on the right may contain thousands of different bacteria, which is an ecological community, right? So you have this entire universe, this massive community of organisms in a single drop of water. And on the flip side, there's about 12 species of tuna fish in the entirety of the Indo-Pacific. So, we're talking about a massive body of water, and we only have 12 species of these particular types of fish. And that poses very different challenges. And I would argue that both of these spatial scales are really difficult for us to work with. And as a matter of fact, our sweet spot often is sort of the scale from a few inches to a couple of kilometers. So think about forests, think about intertidal zones, think about coral reefs. Those are really the scales at which it is really easy for us to observe communities, quantify them, and work with them. Again, as an example, if you wanted to get a handle on the abundance of different tuna fish species in the Indo-Pacific, the entirety of the Indo-Pacific, you might be hard-pressed. But if you want to wander around the intertidal and assess the abundance of different clams and mussels, that might be a lot easier to do. And that scale is also the scale where we often see human impact unfold at its worst. So, for example, we might see a coral reef that is bleaching, we might see a forest that is devastated by fires, or we can count plastics on a beach and see the impact of humans, whereas actually quantifying, for example, the extinction of microbes from drops of water is going to be very, very difficult, and vice versa. It might take a lot of time for us to see whether certain species of tuna fish have disappeared from the Indo-Pacific. And then ecological communities really also underpin the services that ecosystems provide to humans. So if you like eating fruits and veggies, for example, then you will most certainly rely on the communities of pollinators that actually pollinate a lot of these crops. So the composition and biodiversity and abundance of these species is going to be a major determinant of how successful agriculture can be. And likewise, if you like eating fish, then the community composition of, for example, fishes in coastal ecosystems might have a strong bearing on the productivity of that community, how much biomass you can harvest, and the types of nutrients you can consume from these communities. So I would really argue that our planet's heartbeat, if you want to call it that, really goes through ecological communities. They're sort of the nexus, in my opinion, of how things, of how elements move through animals and to us. And as such, they are hugely important. And as ecologists, we're really kind of like physicians. We're sort of make our observations in the world, studying these communities and try to diagnose where the problems are, and then at least argue for making changes that could protect biological life. We're not surgeons. We don't usually intervene too much unless you're sort of in the field of restoration. But I would argue that, you know, communities, animal and plant communities on our planet are some of the most precious things that we have in this universe that really underpins a lot of human well-being. And as a consequence, you'd think that we have a lot of funding for this type of work. That might not be the case. So here's a breakdown of federal funding in billions of dollars. I think this is from 2018 in the United States. So 43 billion have gone through the NIH, National Institute of Health, which is great. Obviously, medical research, very important, trying to cure diseases and help humans survive. Then 18 billion are going to NASA, which space exploration, sure, it's important. I might not be convinced that this is where we should be pumping too many resources, but I'm sure other people would disagree. Then NSF, the National Science Foundation, pulled about 9 billion overall. So this is anything that has to do with science, fundamental science being done in the United States. And of that, environmental science, which encompasses the field of ecology, pulled about $0.8 billion. So let me assure you that within environmental sciences, community ecology is just a drop in the bucket. So you can fathom that less than 1% of federal funding in the sciences is dedicated to studying the life on Earth and our, what I call our most precious resource. And in my opinion, at least, that's a little problematic, but I also obviously have a conflict of interest here in that I would like more money to do research. But that aside, sometimes community ecology can be really easy. So with the stellar sea eagle, for example, we can clearly state it didn't belong where it occurred in Maine, and they got problem solved. I really hope that guy made it back to its original range at some point. But yeah, it can be quite easy. However, this can also get complicated quite quickly. So for example, if we take the surgeonfishes or the family Acanthoria, this is a family of coral reef fishes. Sometimes they're quite nice and beautiful, sometimes they're brown and ugly, but they're quite common, quite abundant on reefs globally, and there's about 85 species across the globe that swim around, and they fulfill different roles. Some of them will eat detritus, some of them will harvest macroalgae, and so on and so forth. And let's just say, as a thought experiment here, that any given reef, you can pick 7 surgeonfish species from your pool of 85. Again, it's just a thought exercise, obviously not realistic, but let's just pretend it is. So you've got 85 species, and you're plopping 7 of them on any given reef. Does anybody want to know the number? Well, the number is of different combinations is 4,935,847,320 possible combinations of 7 species from a pool of 85. So what I'm trying to illustrate here is that community ecology and understanding why certain species occur in certain places and what they do can be quite complicated. And one of the reasons why I think that community ecology is so complicated is something called the mid-domain effect. So I've showed this earlier, basically giving you the different scales of biological organization. And if we assume that processes sort of span a couple of hierarchical steps, then what we'll find is that communities, by sitting at this nexus right here, are where most processes overlap. And that may be one of the reasons why community ecology is so complicated and so complex. And it may be one of the reasons why most of the time in community ecology, we just say, well, it's context dependent. You will see this all the time. Sometimes something happens a certain way and sometimes it doesn't. This context dependency is infamous in community ecology, and if you go to some conferences, you will find that a lot of talks essentially end with, yeah, well, it depends. A lot of what we know about community ecology and the vast body of literature is derived from systems like this. This is a beautiful meadow in Upper Bavaria, nice and green, nice and lush, with a bunch of different species forming this grassland ecosystem. And grasslands are wonderful to study because you can identify a lot of the plants in it. You're working with a manageable spatial scale. Grasses grow really easily as well. They grow fast, so you have a nice manageable temporal scale as well. And then there's nothing wrong with being outside, weeding a sunny little patch somewhere in southern Minnesota and having a mighty fine time. Whereas marine communities can be a lot more hostile. So if you imagine getting in the water right here to study the community on this rocky shore in Northern Ireland, that seems a lot less convenient than frolicking through the meadows somewhere and identifying different grass species. There are a couple of systems in the marine realm that have been studied particularly well. One of them is the intertidal, the main reason here being that we can walk through the intertidal, do our surveys without any assistance. We can see a lot of things, and they're also super dynamic, so very interesting systems to study. And the other one are coral reefs, where a lot of community ecology has been performed, and the reason for that is that coral reefs are just nice to study. You can see things underwater. They're usually shallow. They're usually not too exposed to wave action, and they're pretty. So don't ever underestimate the attractiveness of working on pretty things. So what does that mean overall for marine community ecology? Well, the marine biome is the largest, most interconnected biome on Earth, so there's vast potential for species mixing and dispersal and all sorts of complexities arising from that. It's the most inaccessible habitat we have on Earth. There's a lack of knowledge about basic processes, sparse theoretical concepts to provide general guidance, and empirical evidence mostly comes from different ecosystems, which is phenomenal because that means that there's still so much to explore and discover when it comes to marine communities. It's really a wide open field that is thirsty for knowledge. Okay, so I've talked a lot about species and ecological communities, and I want to go ahead before we finish this lecture and actually define what ecological communities are. Valand in 2016 thus has a definition of community, which is the complete set of organisms belonging to all species, from viruses to microbes, plants and animals, living in a particular place at a particular time. Now, that is a great definition, and if we sort of draw this out, we would consider, we have a couple of predator species, we have a couple of primary consumers or herbivores or detritivores and omnivores that get eaten by the predators, and then we have a whole bunch of plants as well as microbes that sit at the bottom of the food web that will produce a lot of the energy and nutrients that flow through the system and also break them back down when there are feces or carcasses that release energy and nutrients. So that's our ideal world. And if you look at this photo right here, you can imagine that, studying this community from the microbes in the corals to the small fish, to the big fish, the sponges, the shrimps, the crabs, and the sharks that visit would be the best possible thing you could do. It sounds like an absolute winner, but I'm just going to throw this number back out here. 4,935,847,320. Those are the possible combinations of communities you can get from your 85 species of surgeonfishes. So that's just one family of fish. Now you can, vastly expand that to essentially an infinite number, something around 1 million different species, and then try to do the math, and it just becomes completely ridiculous. So what I'm trying to say is that the vast majority of times, studying the entire community from the viruses and microbes up to the top predators is not going to be a logistically feasible thing to do. As a consequence, we often focus on certain sub-communities. And those communities are formed by either similarity or by interactions. Let's talk about interactions first. So one of them would be predator-prey interactions, which is sort of the food web approach. And oftentimes what we would choose to study are these communities of species that are somehow linked trophically across one or two levels. The other one are mutualisms, strong positive interactions. So predation, obviously a negative interaction, especially for the prey. Mutualisms are much more positive, and the prime example for that would be plant pollinator interactions. So again, we can draw this up. So predator-prey interactions might be a predator and herbivore on a plant, a nice community to study, whereas the mutualistic community would be plants and pollinators, for example. For predators and prey, oftentimes they are somewhat comparable ecologically, sort of operating at reasonably similar scales. You can see right here, this is a lizardfish that is eating a very clearly very distressed goby. They're about the same size almost, and obviously therefore are subject to very similar ecological processes. That gets relaxed a little bit when we're talking about things like whales that would feed on krill. So there the spatial scales of existence are quite different, but still, they're both animals. They're somewhat exposed to the same ecological processes. On the other hand, especially mutualistic interactions can operate on very, very different scales, organismally speaking. So if you think about a coral, for example, a coral is an animal that is built by all these tiny little polyps that you can see with their tentacles sticking out. And within these polyps sit these dinoflagellates, zooxanthellae, microscopic organisms that photosynthesize, that are under vastly different ecological constraints and pressures than the coral animal itself. So a coral can live, the whole animal can live for up to 300, 400, 500 years, whereas these tiny microorganisms that live within them obviously have turnover of hours or days or weeks. So vastly different scales of life that come together here to form this mutualistic interaction, which can make it a little more difficult to sort of apply common concepts to these communities. Beyond the interactions, the other sort of guiding principle here is that we can look at species with similar ecology or evolutionary history, which form these assemblages, guilds, or horizontal communities. And one great example are your surgeonfishes from earlier. You can tell by looking at these 6 fish right there, they all kind of look similar. They're all in a reasonably similar size spectrum. They all do reasonably similar things. So they're a great community to study. And that can extend even to, other trophic modes where we can have a goby, butterfly fish, and cleaner fish. So fish at this point, the phylogenetic branch on the tree of life that contains fishes, is our community. There's a shared evolutionary history here that we assume makes these fish similar enough that we can study them under the umbrella of a community. And likewise, we can do this without shared evolutionary history. So if you look at the bottom, we have coral, sponges, bryozoans, and algae. They're very different, evolutionarily speaking. But they compete for the same resource. They all want space on the seafloor, And as a consequence, they interact in so many different ways that they form a really nice community for us to study. And that's it for today. The first homework is to draw an ecological community of choice. I've just exposed you a little bit to what communities are and what makes them ecological communities. So I'm asking you to draw something, any medium or any approach is fine. You can draw stick figures if you don't feel like you're particularly talented, or you can do scientific illustrations, you can do watercolors, you can do anything you want. Just draw a community and tell me a little bit about why you drew it and what makes it an ecological community. And that is due because it's MLK Day next week. That will be due on Monday, January 27th. The second homework, you might want to start with this one, is to watch the R tutorial for Thursday. I will demonstrate this in a second, just to give you an idea of what to do. There are two tutorials. There are these videos that I've published, and just watch them, and then we will do the exercises in class. So the next class is at 1 p.m. on Thursday. I will see you there. Have a great Monday, and I look forward to seeing you later this week.